We are to start the last, but not the least, session of the day with personalized or precision medicine. Team is led by Daisy, so please.
So we're gonna talk about network personalized medicine, and we're gonna focus mostly in inside clinics, so mostly using omics and image data. And Don is gonna be doing the introduction.
Thank you.
So first we have the technological. The bottom is the pointer?
Yep.
Yeah, okay, great.
And the other ones will change as well.
Okay, perfect. All right. So thank you, and thanks for Daisy for volunteering to wrangle this particular group of cats, and thanks to the organizers as well for having us here. This has been really fun. So
The other ones will change as well.
the other, oh, the whole other thing? No. Ah,
Green button.
there, that's a button. The user interface on this, okay. So actually, this image really cracks me up in part because, all right, so we started this off and said, okay, what do we actually mean by precision medicine? And it turns out that most of the papers don't really agree on all of that. So we're gonna talk about that for a couple minutes. But even just in terms of why are we here and why are we thinking about precision medicine? This is a really amusing timeline because actually there's been an awful lot of progress in medicine between the beginning of the Enlightenment and the 1990s. But one of the things that I think is important and missing from this is that the standardization of concept of a randomized clinical trial and what kinds of evidence you need to draw conclusions from that kind of intervention didn't really happen until the 1940s, which I was really surprised by. It's quite late. I think the other thing that cracks me up is this concept of evidence-based medicine, which is, I mean, it's a big deal at Tufts. We have a whole, clinical department that's really well known for that. And I always sort of hear that term and think, so what were we doing before that? Isn't the whole goal of medicine since the dawn of time to treat the patient in front of you? And isn't that kind of what precision medicine is all about? So, I mean, yes, it is. And we've known for many years that there are different blood types and we should, not mix them. That, for example, women have different heart attack symptoms than men. So we've done this sort of patient grouping informally for a long time. I think what really is driving the precision medicine initiatives is that all of a sudden there became, in the 90s, there suddenly was enough data to start to say, I'm not just going to treat patients based on my prior experience of the people who walked into my lab or clinic most recently, but I'm going to try to define what the group is empirically and then statistically gather all the data and look at all of it and make decisions based on that. And then that, as the data growth has increased in recent years, we've started to become more and more able to integrate, omics data and online, electronic health records and other kinds of data in a much larger, more systematic way, and begin to say, let's really try to tailor things to individual people. And so that's kind of how we got to the point where this even was on the president's radar of, okay, we're actually going to invest in this precision medicine concept as a thing. And again, when I think about precision medicine, I'm mostly focusing really on therapeutics. Okay, I want to know how to treat a patient. But there are a lot of other things you can do with this sort of information, right? You can certainly improve and refine diagnoses or disease characteristics or definitions. You can try to develop new drugs for specific populations that you might not have known existed. You can even try to develop drugs for individuals. Right, and so all of that still falls under this realm of precision medicine. So what are we really trying to do? Right, so the most common thing that we're trying to do, probably what I think the president was mostly thinking of, is this sort of notion of targeted therapies to groups. Right, and I think nowhere is this more obvious than in the way it's revolutionized cancer in recent years. I just put non-small cell lung cancer up there because it's something I know a little about, and it's a tremendously devastating disease. It's very common. It strikes people relatively young. We don't diagnose it until it's usually incurable. And the life expectancy and quality of life upon a diagnosis is dismal, typically. And what has happened in recent years, it's still dismal, but it's less dismal, and the reason for that is that we have identified a number of different causative mutations or families of mutations that are targetable by specific drugs that didn't exist 10 years ago. And these have a tendency not only to improve survival, but also to dramatically improve quality of life, because the alternative is these platinum-based doublet chemotherapies that are like everybody's nightmare of what chemotherapy is, right? And so most of these drugs are better tolerated. So the idea that we can say, okay, you have an EGFR mutation, you're getting this particular drug, and you have a different mutation, you're getting this other drug, is considered precision medicine, even though it's really hitting large groups of people. And so that's really made a big difference. Then there's another whole group of people who, when they think about precision medicine, are thinking about something entirely different, like an individual clinical trial, right, or a drug developed for an individual patient. And a lot of these kinds of things have come up, particularly in the realm of rare pediatric genetic disorders. So the sort of maybe most famous example of this is this drug Millicen that was created for Mila, who had this rare form of Batten's disease. Tim Yu from Children's talks about the process he went through to do this. I mean, sequencing and identifying the causative mutation was not that revolutionary or different from what everybody in here is familiar with. Getting the FDA to run a clinical trial for one kid was a process. And they were able to do it. They were able to do it because her family had access to resources that the average family does not. This is not a scalable thing, and I've talked to Tim a little about this. And his hope is, well, okay, maybe eventually it will get to be scalable, but I don't think this is going to be the way we normally do medicine. But they were able to convince the FDA to let them develop and try a specific therapeutic for a specific patient. Some of you may also be familiar with the story of Matt Might, who was a programming languages researcher until he had a son with another rare genetic mutation that led to a deficiency in the NGLY1 gene. And his way of dealing with having an N of 1 was actually kind of creative. It was to use social media to make N bigger. So he reached out to the world and found, tens of other people who had a similar disorder. And he's done a lot of drug repositioning and repurposing that has been somewhat helpful in staving off some issues with that disorder, but there's more to be done there as well. So what I really think of as the Holy Grail is this sort of patient-specific but not FDA-involving process, ideally, where instead of developing something new, you can say, okay, for each patient in this room, which is the antidepressant that would be most appropriate if they needed one, right? So we have a choice of a bunch of different things. What's going to work for you? And maybe that does include some external data like Fitbit data, which I put in there because it's been proposed as sort of outside of the clinic. But a lot of the stuff that we are going to focus on clinical data because that's mostly what this subgroup does. But I think there are a lot of people in the room who are working on integrating extra clinical data with clinical data, and I would like to keep the discussion going in that direction somewhat too. So just to give a couple of examples, since I haven't talked about networks at all yet, and this is a network workshop, right? There are a lot of, it's actually interesting, there are relatively fewer than I would have expected sort of home run hits where we're really doing precision medicine targeted at individuals using networks in a sort of integrated way. But when I sort of looked at the most recent research, a bunch of things did jump out at me that were doing that. And a lot of them are building patient-specific networks. So this is one that was just on the top of my pile of science journals in my office. Came out a couple weeks ago from Weill Cornell. And basically the idea, and I'm summarizing a huge science paper in 30 seconds, so I'm omitting a lot, is that they looked at attack-seq data and RNA-seq data over a bunch of different kinds of samples, integrated them from prostate cancer samples, and found patient-specific networks, and then tried to identify the transcription factors that were driving the individual, or sort of at the core of a lot of the changes in those patients, and then clustered them. And what they found was that they reproduced a lot of known groups of prostate tumors, but also identified this new stem cell-like group, which is really cool. Somewhere in there's right, because so there's a transcription factor at the core of that binds to some other proteins that are actually where there are actually known therapeutics that target them, right? So they're proposing trials to use those inhibitors for that subclass of patients, right? So this is another sort of approach to targeted therapies, but was done with these individual patient-specific networks. That are pretty cool. And similarly, there was another very recent one on ulcerative colitis that, again, was looking more at SNPs, but again, built patient-specific networks. And then instead of working on the individual patient, sort of said, okay, again, we're going to find big common subgroups of those. Right, so there are different ways of doing this, but you can begin to see how you could both use the group to find immediate targeted things and use the individual to maybe make things even more personalized. So with that, I just want to sort of give you these are the notes I jotted as the outline, and we never fleshed it out. But the idea is that we're going to have some discussion. I think the first biggest question really is, how do we integrate all of these kinds of data, right? We have so many different things. How do we put things together? Some of them are, we've talked a little about data integration. We've talked a little about the scale of algorithms. But this is going to be an issue because some of these things are at very different scales than others, and the methods you use don't necessarily translate one to another. We're going to talk about noise. In the noise section, we're also going to talk about imaging, which is something that has kind of not been a focus at this meeting, and I think could be another whole meeting. And we'll talk about some applications as well in this field, and then what comes next, which is always the big question. So I think with that, I will hand this off.
That's a great introduction. So yesterday we have excellent session to discuss heterogeneous data integration. So in precision medicine, the situation is different because in yesterday's session, most of we talk about integrated heterogeneous biological network. Oh, sorry. Yeah, we talk about how to integrate a heterogeneous biological network. Then our aim to do node classification or link prediction. In precision medicine, most time we have for the data we have they are sample based. So basically we study the individual patient, individual subject who has both like a genotype, phenotype. So across the different subject, we hope to identify the important biomarker to develop like a drug and provide a treatment plan. So since both genotype and phenotype provide different views for human biological mechanism, so they complement to each other, can help us understand better for human conditions. So definitely there is a great need to integrate multi-scale biological, biomedical, like especially for genotype, phenotype data for the precision medicine study. However, there are also computational challenges. For example, when we integrate heterogeneous multi-scale data, so we need to consider the interaction between them. So the interaction is not obvious because it's different to the biological network. We can find a protein-protein interaction, gene interaction. So for the genotype phenotypes, so the link between genotype phenotype, they exist but not obvious. So we need to uncover such underlying latent interactions. And meanwhile, we have lots of the data can be formulated either like a vector or the graph, like a pathway network. So how can we integrate all those data? They are also very challenging. And the third problem I will talk about is in our large cohort study, we have both genotype, phenotype the data. We can integrate them like train AI models. However, When we apply the model to the real world setting, so in small clinic or some other hospitals, they only have single model data, like either genetic or image. Can we train using the multi-scale heterogeneous data train a better model to help single model data prediction? It also is a very interesting and challenging problem. So I'll start with my previous work to integrate the phenotype genotype data. Like for example, in complex brain disorder study, so we have like MRI imaging, we have functional MRI, TTI, we also have brain networks, and meanwhile we have the genetic data and also protein data. So we want to integrate all those genotype, phenotype together to predict like a cognitive decline or some other mental health problem. So one key step to identify the biomarker, potential biomarker, especially from the genotype side, because we hope to eventually develop a drug like to do the treatment for Alzheimer's disease. So when we integrate such genotype phenotypes, we hope to learn the interaction between these genotype phenotypes. We need to consider typically we call the global importance. That means from a global point of view, some data modality will be more powerful in prediction tasks. And meanwhile, we need to consider the local importance. That means even one modality is not very powerful in prediction. However, for individual markers, they could be very strong signal in the prediction. So we need to consider the overall for modality point of view and also individual's feature point of view. So I started my early work at ISMB 2013. So we first developed using the structured sparsity-induced norm-based method to learn such global and local importance. For example, here we is there any laser? On the bottom.
Yeah.
Oh, thank you. It's so smart. So we impose first we have a randomization called group one norm. So L1 norm means we impose sparsity between the image data modalities like genetics, phenotypes. So for every subject, they will have the every row will be one subject. Each subject will have some MR data, TTI data, network data, and SMP data. And each subject will have multi-scale heterogeneous data. And some subjects belong to like Alzheimer's disease. Some subjects belong to like MCI or control group. So first, group one norm will impose for every modality for individual patients, the individual samples, the subject, we group them together. Within the group, we impose L2 norm. Then between groups, we impose the L1 norm. Such that we do the data-driven approach. We will do train the model. So the L1 norm will have the competition between different data modalities, between different subjects. So the less important modality and for subject will be subtract, like the shadow color. The important one will be pop up, so we can enhance the strong signal for the important modality. That's for global importance. That's for global importance. Meanwhile, we are the second term we call it 2-1 norm. So we consider the local importance for individual feature. Even for one modality, the whole modality may not be very strong, but individual markers they could be strong. So we cross at a perpendicular direction with L2-1 norms. Within the group, we impose L2 norm. Between the group, we impose L1 norm. Such that for individual features, if they are important, during the model training process, they will also be enhanced. So through these two regularization, we can do the data-driven approach to simultaneously learn the interaction between individual features and between the modalities, and to get a strong signal to find biomarkers. Here I didn't actually because for genetic data we often have the like a gene interaction network or protein interaction network. We can add the like Laplacian embedding at the W transpose G the graph, the Laplace of the graph, to embed using the gene interaction information during the subject-based training model. So incorporate the existing knowledge. So through this learning, we can find do the integration for multi-scale data. So later we improved this model in our the KDD paper around 2016. So we also apply the same approach to the Drosophila gene expression image annotation. So for Drosophila images, they have for every embryo they have the different nodes. The different annotations. For different term we often need to automatically generate them. So in one of my project we use different there are different computer vision like shift different different descriptor. After we get this descriptor, but they only capture partial of local information from the image. Then we can apply our approach to the integration to predict those terms and the states to the annotation for the Drosophila embryo image. So this is for the standard multi-scale data integration. Like in our brain study, we also beside the like MRI genetic data, we also have the brain networks. Like for every subject, we can scan the brain, like either scan DTI or get structured data or get do the function MRI to get the function networks. And for every subject, we have one brain network. Meanwhile, because when we do the extract structure network, we have multiple ways, and every way is designed from different underlying biophysics mechanism. So that means for every patient, actually, we can generate multiple brain networks. So in our study, we hope to integrate such networks to do the prediction, to identify the marker, the network marker, which will help us associate to like Alzheimer's disease, MCI, or control. So then this problem become the integration of multiple brain network for each subject, and then we have many subjects. So that's become the brain network integration. So we conduct study to design a model using graph convolutional neural network to design a model to integrate the multi-scale brain networks, then to predict that we conduct on the Parkinson's disease to study the Parkinson's progression for the mental health. So when we developed this method around 2018, at that time the pooling method for graph convolutional neural network is not very popular yet. So it was not well designed. So we designed a adaptive pooling method for graph convolutional neural network. When we designed the pooling method, the key idea is we hope to when we do the pooling, we merge the local structure of graph, because pooling on graph is different on image. Images have clear structure. For graph, we don't have, so we need to find the local information, aggregate them. Meanwhile, we keep we call the loyalty. That means when we reduce the graph structure to do the pooling, we still keep the main structure of the whole graph, the whole global view. Meanwhile, we hope make sure the competition is very fast, cannot be slow. So we design a adaptive pooling method. So when we do adaptive pooling, when we do the graph, we minimize between layers their global structures to keep the loyalty, to keep the overall structure for the graph. And meanwhile, we added this the L1 regularization to make the sparse is similar to the classroom results to find the local sparse connection between the graph, do the pooling, merge and node together. So then based on adaptive pooling, we design this multi-scale brain network integration for every subject. Every subject has a bunch of brain networks. They extract from different DTI methods. We could also add a functional MRI network. Then the adaptive pooling do two ways. The first step to extract extract the node to the clustering aggregate using the L1 regularization. The second step, we do the distillation, shrink the network to a small ones. So after multiple step poolings, we can put them together to the prediction. So, well, at that time, the contrastive loss was not hard yet. So actually, this step later can add contrastive loss to do the first integration to predict to the final prediction. So beside the integrated multi-scale genotype, phenotype data and the brain network data, in recent years we also consider. So in large cohort, we have image and the genetic data. For example, for AMD, age-related macular degeneration study. So we have both image and phenotype and genotypes. In the large cohort, we can train the models to the prediction. However, if we apply this model into the real-world setting, we often only have like a single model data. So we hope to utilize multi-model training to train a good model. It will not just work for the multi-scale data, also work for single model data. So the main idea is that this paper was presented last month at the Recon. So this mathematics we consider, we train a multi-scale model, integrate both the genetic data and the image data. And then we do the knowledge distillation. We distill knowledge to help the training of the single modality model. Then do the prediction. So then the single model the single modality data, we can do the individual prediction in the inference state. Then we can using the multimodal data to help single model training get a better result. The main architecture is like this. So like for image side, we use the focal loss. Focal loss is a sparse is a L1 based loss. It's better for noisy image. And then we do the standard feed forward network for genetic data. Then the integrated, if we use the Riemannian time model to do the non-Euclidean embedding, then to integrate the data to do the prediction. So up to the prediction, in the training step, the single model and the multi-model, their knowledge will transfer to each other. They help generate the genetic data to the prediction. To make it pull the single model distribution to the multi-model distribution. So after training, we can get rid of this, for example, get rid of one type of data, like genetic data. Then using the phenotype data to do the prediction. So for the real setting. And because this network, if we have the network input as input, for such like the feed forward network can be replaced by a graph, the graph, the GNN or GCN to deal with the graph input. So yeah, that's for some challenging work in multi-scale integration. Yes, I kind of design it.
Thank you. Okay, so I think the noise in data has been discussed quite a bit in other form. So today I'm going to just focus on biomedical image and to try to mention some of the issues arise. So we all know that, and I think I should stay here. Yeah. Already, I got only two hands. I get my feet. We need more technology. Okay, so we should all know that when we have data, image data, and the people try to process them, right? So the common algorithms are just like CNN, GNN, transformers, these kind of things. And the purpose, of course, related to biomedical medicines, like diagnosis, and we discover knowledges. So these are the common practice. And then, of course, biological network arise almost naturally in the process of image processing, biomedical image processing. But one thing I should mention is that unlike a natural scene image, which are mostly two-dimensional, biomedical images, they are much more challenging. So they can be 2D, 3D, even 4Ds when we have time. And the size can be very large. Some of the data we have, for example, easily will be like one single 3D image is 2000 by 2000 by 2000. And then you plus time. It's huge. It's very difficult to process. So they raise very different challenge comparing to just common computer vision tasks in natural scene image. And often the number of objects in biomedical imaging is very large. We can have like millions of objects, not like in a natural scene image we have dozens, a few. So they raise very different challenge. So of course, the image, they can come from many different sources. They can come from basic biomedical research, come from clinic and outside of clinic. For example, people nowadays use a cell phone to take a photo or an image of their skin, and they can send to the clinic and they ask for some kind of answers. And there are situations like the battlefield, the soldiers there, they get handheld device, they get their image, and there will be no doctors on their site. And there will be natural disasters, people go to rescue, and we get image. So there are lots of sources of image, and we need to study them. And then, so there are lots of biological network arising in this kind of scenario. This is an image from, of course, founders. But this is the question raised by medical researchers to us. And they said, the eye image is some of the most easily accessible image, no damage to human health. So several sources of image are very easily obtainable, like the skin image, the eye image. But they said, we want to use this kind of image to study other very important disease, like the heart disease, the brain disease. I was surprised. I said, can we use eye image to study and to predict that kind of disease? They said, yes. We can collect lots of data, and we think this is a way for us to understand some of the very difficult disease without very invasive way to generate image. So for example, in this situation, you can define network and try to understand what the network is trying to tell us. For example, the blood pressures and the change of the network. So there are lots of stories. So let's try to look at what information an image can give us. Image information can be viewed at different levels. Normally, people focus on the appearance levels. What is appear to me? What are the objects? So at this level, you can view that image. They are in the pixel space. So there can be a very large number of pixels, but they are kind of quite low levels comparing to other levels, as I will discuss. And people can use CNN, the kind of method to deal with them, and we can study classification, segmentation, detection, tracking, registration, those kind of problems. But they have drawback. So the drawback is that the field of view of this kind of method, like CNN, there's a trade-off between the size of the field of view and the patch resolutions. And they may not really give you lots of information at the higher level, like the tissue levels. They just give you lots of pixel level information. So then people try to introduce, so the image level, basically people ask a question, what are the objects? So what do we see? But sooner or later, people start to ask the question and say, what are the relations among those objects? So graph is one way to address the question on what are the relations. Graph basically focus on the pairwise information. So for example, you may say you have a cell graph. So the cell graph is like the nodes are the cells, the relations, possibly we can assume there are some interactions between cells when their distance are not too far away. Then we can define cell graph, or we can define the gene ontology graph. And then, so in this example, we can define some of the cell relations. And the tool for processing network is like GNN, GCN. So they are very good at exploiting pairwise relation. However, relations are not only limited to pairwise. So there are other type informations. So they still have a local view. So we propose to explore the topologies of image data. So that's what our group is trying to do at the recent time. The topologies, they are capable to capture higher level information and the relations. For example, what are the distributions? So this kind of relations are important because in one of our current studies, the biological researchers always ask question, what are the tumor microenvironments? Then I ask them, what do you mean by tumor microenvironments? Can you tell me something I can use to identify tumor microenvironment? They say no. I said, can we define or specify some kind of fingerprint so that when I match the fingerprint, then I will say this is a likely site for tumor microenvironment. They said, we don't know about that, but I can give you data, right? I can give you data, such as what? You will see some of these data, and like a very complex, a three-dimensional brain image with lots of cells, tissues, tumors, and we are supposed to be able to specify something for them. So this is more than the simple notion called biomarkers. Biomarker to me is something like simple features, but this is more complex. So we try to capture at a higher level the topologies. So the tool, one kind of tool to use is the persistent homologies, but there are other tools. So I just use that as an example. So this is a good tool to capture higher level structures, but the drawback is that they often ignore the low level details. And that's not good to ignore low level detail. So what do? We combine the multiple levels together. So this is an overview. So you can see the current approach. They have a CNN, that's not CNN for image level, and then we have graph level, GNN. So we have the topology level. So we look at the literatures. You can see that people already use image plus graph approach, and graph plus topology, image plus topology, but combining all them together. So this is the approach we are trying to explore. So here's an example. In this particular study, we try to look at the pathology image, and we try to classify the image based on the tumor grade. So which level of the tumors, grade 1, 2, 3. So in this situation, we have the low level image information, and then we can use graph to capture the local cell interactions. But then we gradually go into different scales of cell group, and you may also call them clusters. But in terms of the topology, it is not a fixed notion of clustering we are talking about. For example, if you use the density-based clustering, you see, okay, there's a dense parameters, and if you use that, you compute clusters. But in topology, there is a function called filter functions. And if the filter function is the densities, it's capture different scales as the function value changes, different scale of the clusters. And that gave a tree structures, and we call the filter tree. We introduced the structure of filter tree. And then we combine these multiple levels informations together, And then we did our preliminary studies, and you can see that the result improved on the state of the art by quite a bit. So what I would like to mention, a very important challenge in biomedical image processing, which is the labelings. So usually people have to label a lot in order to have data for the training of the deep learning models. But labeling is a very challenging task. So these are some of the images I just mentioned for study the brain tumor microenvironment. So these are called the glia cells, different type of glia cells. They are in three-dimensional space. You can see how dense they are in 3D space and how complex their shapes is. So to label those, sometimes my students have a great deal of trouble. One summer, 2 students labeled 2 weeks. 2 weeks all the time. And at the end of these 2 weeks, I asked them how many cells they label in 3D. They said a dozen. It's very hard to do. So these are some of the real image that we got. And this is glia cells. They're just like trees when they are far away from tumors. But when they become very close to tumors, they change their shape. So if you look at this shape closely, I think they're like dementors in the Harry Potter movies. So why they change the shape in this manner? Because in that shape, they can touch each other, build a network. But when they close to tumor, in order to play their immune function, they want to insert themselves into the tumors. So they change the shape so that they are able to do that. But in that situation, we did a hard time labeling the normal one. When they look at the close side of the cells, all the labeling is useless. Okay, do it again. And then tomorrow, the biologists will change the experiment. We use a different line of mouse, and we use a different line of cancers, and you just continue to labels. And in 3D space, it's very, very difficult. So this is a big, big problem. So we developed a whole bunch of techniques for reducing the amount of labels. So for example, if you really look at your image data, you'll see that some of the data, for example, they are the rare cases, but they are the case we really care because they are about disease. So we want people to label those. But for those common examples, they are very similar to one another. And you only need to label a few of them. They are representative. So in this way, you don't have to label everything. In fact, our study shows that for some of the even very difficult image, you only need to label about 3% instead of 100%. So you can aggressively reduce the amount of label and to achieve very good result. And another problem that commonly people deal with is noise, blurriness. So these are the image data from our collaborators. They did the experiment. They said we cannot see a thin, right? We don't know what's going on. They just kept the data, put it away. After they kill all this mouse and all the experiment, they don't see anything. They just put the image away. And we said, we may be able to deal with it for you. So we fought it for a long time. And eventually we cleared the image for them. But compare the approach that currently used in the state of the art. Usually this kind of a so-called image restoration, and they require to clear image plus the unclear image because they want the deep learning to learn how to map the unclear one to the clear one. So we asked the biologists and say, give us some clear image. They said, if I have the clear image, why do I need you, right? I finished it. I finished my analysis. So we have no clear image. We have no idea what's going on. Just do it. So we developed a method that we require 0 high quality image. And also many of the approach assume that you know lots of the parameters like the point spread functions when they create the image using the device. And we assume 0 such knowledge. All we need is that just give us the dirty image, we will be able to clear it for you. So that's what we have done so far. And in my short time, I will Yeah, there is a.
So in this part of the presentation, we will move to application of the algorithms that we discussed yesterday, the graph theoretical type of algorithms. So I'm going to start with the first application, namely looking at application of various type of disease-associated models that we discussed previously, various methods to identify those models. So how that relates to personal medicine? Of course, we appreciate that most of the complex diseases are studied from the perspective of dysregulated pathways rather than dysregulated genes. So of course, identification of this disease-associated models is important for understanding of the disease, but also is important for understanding heterogeneity of the disease, which allows us to identify disease subtypes. And then from the perspective of the individuals, it helps us to assign individuals to a given subtype. So I will start with various type of disease-associated models and giving quick examples where they are being applied. Of course, many people in this group have their own application, and this list, could be much longer. So the first type of models is the classical model based on the mutations, where we are given the mutated set of genes across many different individuals. We map those mutations on the protein-protein interaction network, and then based on this mapping, we try to extend the known the genes that are the subnetworks that are defined by observed mutation to include mutations that may not have been observed, but based on the network relationships are predicted to be also related to the disease. Now, there's of course many different algorithms to do that, and be that using shortest path or diffusion method and so on. I'm not going to spend more time on this one. I just want to move on the different type of models. These models I would refer to as the information propagation models. So what we have here now, a setting is quite different. We have information about association of mutations with phenotype and, like we say, expression level. So that would be typically equatorial type of data. And so we know that there are certain mutations in certain genes, yellow here, are being correlated with the changes of the expression of the target red genes. So we would like to understand how those mutations in the genome propagate to influence the expression of a gene that's observed to be associated, whose expression dysregulation is observed to be associated with manifestation of a given disease. Again, there's a number of methods to identify this disease propagation, this information propagation models. One class is based on the shortest path. So what are the shortest paths from each of those mutated genes to the target disease-associated genes? That may be Steiner tree type of approaches. And the second group of methods is more based on network flow type of analysis. So the difference is really what you really think about what is the proper way to model the propagation of the perturbation in a given node to the perturbed node. Is that like a cascade of events, or is rather broader perturbations that perturb broader networks? So this method here is developed by my group. So in this case, we were working with glioblastoma multiforme, and the perturbation for glioblastoma multiforme are usually copy number variation. So those lead indirectly to expression changes. Expression changes do not form this kind of cascade of event, but rather a broader dysregulated pathways. So what we did here was simple, use also expression data to say, okay, if I have 2 nodes, how likely this edge is being used for propagation of the signal. And so we model this electric circuit, putting a big resistance if the 2 nodes, expression of these 2 nodes is completely uncorrelated, and very small resistance if there is a correlation. So either way, in this way we will have, rather than having, a tree-like structure, we have kind of subnetwork structures. However, either way, what both methods look at next is also looking what are the genes, or perhaps in our case we look what are the pathways that were common among many of those gene genetic perturbation, expression, and perturbation association. Okay, the next type of models are the models related to differential gene expression. There's again a number of approaches that utilize differential gene expression. So generally you will have the controls and you will have the disease patients, and for each of them you can see what's the difference between in the expression between disease and control. And then 2 basic groups of approaches, and then this one is a little bit more general. We're going beyond the expression model only, but 2 basic group of approaches would be the first one more similar to the mutated network, where we score each of the genes in terms of how the differential expression, and then use the network information to extend that knowledge to include and score larger models. This can be done, this type of approach can be done where we even have a small set of patients. If you have a large set of patients, then you can utilize the fact that the set of patients large to look at the correlation between expression, and then use also what we call the correlation-based method, which also information which edges are correlated, and identify more correlation-based approaches. And the third here method is based on the extension of the idea of set cover in the graph theory, and is trying, it's not only used in, it's used in actual many applications. Many people here in the room were using this type of approaches, where we try to associate, bring associating with genotype and phenotype. So here you have patients on the top row. On the bottom you have protein-protein interaction networks, and you, for every patient, you have some information which genes are perhaps differentially expressed or maybe mutated depending on the application. And what you are trying to find is find a set of models so that you can somehow optimal set of models, that those set of models can explain all the individual patients. So then again, that's typically would be formulated in some sort of optimization approach. Depending on the disease, you may have preconception. For example, for cancer, you would expect that there will be every patient will have several dysregulated genes. Maybe for other diseases, that number will be bigger. So all those 3 methods actually requires that you have protein-protein interaction network, and you kind of superimpose your dysregulated genes or potential dysregulated genes or the networks that you are trying to identify on this network. Now, this is not always the case. So here is a slide from Daisy's work, where they observe in that various various patients may have differentially expressed various subnetworks, and those subnetworks are here defined based on expression correlation. So this is of course also observed very, very clearly also in cancer, and in fact many of the first attempts to disease classification are really based on this differential type of expression. I'm going to come back to this in a moment. So actually, okay. So this is one example. So where we start by, a so-called patient similarity network that can be really built based exactly on this expression similarity method as previously. And then we would like to use this patient similarity network to, first of all, identifying the subclasses of the disease, but also to identify features that underline those diseases in addition to having similar expression. So one way to do it is to use hierarchical topic model. We haven't been talking about topic models yet in this meeting, so those are familiar to many people, I'm sure, in the audience. But just let me briefly summarize that those are models that are borrowed from the natural language processor, where you assume that you have a set of documents. If document is described by the sequence of words, and you know that those documents are discussing a certain number of topics. You would like to identify those topics. We define the topics as the distribution of words. So you would like to identify the topics, and for every document, tell what mixture of topics this document is talking about. How that translates to the diseases? We think about patient as a document. The words of these documents are all the features patient may have, and this provides a wonderful way of combining various types of features. That could be mutation, that would be copy number variation, methylation, whatever information you know about this patient. We don't use expression because we use expression to build the initial network. So we would like to identify the disease subtypes as those topics, and for every patient to identify what subtype this patient might be, but we understand that patients may not have clearly defined subtypes, but maybe some mixture, some of two subtypes or more. So we use, in addition, the hierarchical topic model refers to the fact that we also respect the fact that we have some similarity between patients, which, I think I'm missing, here, similarity between the patients. So the method infers now the topic, so that if two patients are similar, they are expected to be explained by similar mixtures of topics. So that worked very nicely we apply it to a couple of cancer classification tasks. And also, once you have a topic model, if you have new patients, you can identify, what is the, topic of these patients. The second way that I wanted to, second example of utilizing patient similarity network is from this study of Wang et al. From Anna's Goldenberg group. So they use patient similarity networks to fuse different type of data. So what they started with is they started assume that we have patients, and we may have expression data, methylation data, and different type of data that may be noisy, that may be missing the information. They for each of this type of data, they have the patient similarity network based on this data. So here, say, the patient similarity based on expression, patient similarity based on methylation, and then they have a nice iterative algorithms that fuse those different network representation together to come with patient similarity networks that is being defined by all those different modalities. Okay. Then going back to gene models associated with the diseases, I wanted to say a few words about finding gene models associated with drug response, and then Daisy will say more about drug response too. So let's very quickly, very briefly here. So in this situation, we have again like in related to kind of genotype phenotype type of association for every drug and for every patient, we can see whether, in this case actually cell line, what is the response of a given cell line to the drug. So you can have all the scale of responses being positive, that the cell is being killed, being negative, cell is actually growing even more happily. And then we also have for each of the cell line, we know the genetic perturbations that those cell line harbors, those are cancer cell lines. And then we can superimpose them on the protein-protein interaction networks. And then similarly, as in the spirit of this set cover types of approaches, we will try to identify the models that are associated with the phenotype. A new, of course, technical difficulties, technical challenge that now we don't have like discrete phenotype, but we have continuous phenotypes. But still, we can use optimization type of protocols to identify those subnetworks, mutated subnetworks with each drug response. This again, once you have those subnetworks, that helps you to decide if you have given patients, predict what will be the drug response to this individual. So that works quite well. But also as a side bonus, we kind of identify the subnetworks with certain pattern of drug response. We can look at all those subnetworks, all this drug response, and try to look at complementary patterns. So without going into details how we look at this complementary pattern, it's easy to convince that we will be able to find complementary patterns, and those complementary patterns are likely to suggest the drug combinations. And that was also worked quite well. We identify many drug combinations that have been tried in clinical trials. And with that, go.
So another thing that is quite important before we go back to drug repurposing is understanding a little bit of disease comorbidity. Because patients that they have one disease, so why is it important, right? So one thing is that you can define the path of like what's the next disease that a patient can have. So let's say the patient has high cholesterol. Can this patient go for have a heart attack or some kind of stroke or some other diseases? And being able to understand the disease progression, what are the diseases this patient can have, can help us identifying what kind of treatment would be the best treatment for that particular person or that particular group of people. Oh, and I don't know. Yeah. So we know the diseases that they are similar to each other given their disease modules. They are often located in similar regions in the protein-protein interaction, or they also have similar co-expression levels to each other. So here we have, for example, rheumatoid arthritis is very similar. It's very close to lupus, and about 60, patient that has RA has 6 more chances of having lupus later. So given that, we can always identify which would be the next diseases that a patient can have, and I already discussed this. That the treatments can also be overlapping. So that goes back to drug repurposing. So given that, I got lost in translation here. So one big example that we always have for drug repurposing is the case of sildenafil that was first approved to treat CAD and hypertension, but was in the clinical trials, they found out that patients were having side effects, so unexpected erection. And then they started trying the same drug for other diseases. And there are different ways. I keep playing the thing. There are different ways you can use drug repurposing. One of them is starting from the disease module, from the disease, and trying to identify where the targets from drugs would hit the neighborhood of that particular disease. And here's an example on COVID. So here's the disease module from COVID. So those are targets that SARS hit in the human body. And we try to identify which were the drugs that they were surrounding it. And one thing that is quite important to notice that disease modules, they are also tissue specific. And in a similar way, drugs would also be tissue specific. They're gonna work more like in a context specific than overall. There are plenty of methods for defining drug repurposing methodologies, and I'm definitely not gonna get into details of many of those. But I'm just gonna point three main categories that we have. So the first one is network-based. So you have a disease module, and then you have genes that they are targeted by some kind of drug, and you try to get them as close as possible. So you basically use an average shortest path here. A second mainly used methodologies based on network diffusion, where you're gonna try to diffuse your disease targets to your drug targets and try to identify which one are the closest or which have the highest probability of getting to hit the targets. And we also have the AI-based models, which, for example, they use GNNs and other methodologies. And I keep pressing the wrong ones. And then, thank you.
So, yeah, I guess given time, we'll do this. So there are a bunch of things that I think I'd like to steer the discussion towards. And I guess the first of these actually came up a little bit yesterday, right? It's the question of how do we evaluate when we're doing this well or not, right? And there's two parts to that in a precision medicine network-based model, first of which we did talk about, right? Which is how do you know a network that you're using is actually the right network? And this is, I mean, there are some good comments on this from a paper out of Russ Altman's group. But the upshot of this is a lot of the time the standard way of validating a network in this field is to remove parts of it and try to reconstruct parts of it. And that raises the question of how meaningful is it to actually use a partial network to do that sort of work? Do we really believe that that's a good thing? And how else could we do this? Do we actually have gold standard data that we know would be right? If we don't, one of the possibilities is to use simulation. Now, I say this with some trepidation because I'm very nervous about our ability to simulate particularly like human clinical data. I think we do a horrendous job of this. Other extra clinical, if I can use that word for outside of the clinic, other kinds of data, maybe we simulate with different degrees of fidelity. But the good thing about simulation is that you can use it in specific ways carefully to test specific properties of methods. So that might be something, again, you know, all models are wrong, but maybe you can come up with a model that has something useful to say. So maybe there are ways of doing this there. But there's a bigger question in the precision medicine application, which is how on earth, you know, if we're doing precision treatment, how do you run that experiment, right? The ideal thing is you clone a person at their current stage of life and you try them on 16 different drugs and you see which one works. And of course, this is not very feasible. And, you know, cloning them, doing the sort of animal study equivalent where you do this in mice really isn't relevant to the kinds of things we're trying to do. So what does it mean when you want to talk about validation in a precision medicine context? How do you know if this is right? Well, maybe you look at a population of people for whom you make recommendations and try to say that we did at least as well as the standard of care in terms of outcomes. And then there are all sorts of questions about bias and context and, you know, and even what are we trying to validate? Are we trying to validate the algorithms? Are we trying to validate sort of the predictions? So there are a lot of really crucial questions in this kind of, how do we know if we're doing this well field? And ideally, in order to do that, even that kind of sort of numerical study, you'd have to test a largish population and make recommendations for a large population. And that's, I mean, a little bit, the dollar icon is deceptive because there's also a time and a human cost to all of these things, and we can't really just treat them like experiments. So the other point that I wanted to bring up, and this gets expanded multifold when we get to data that is out in the wild that is not clinical data, is this sort of whole question that we could run a whole other workshop on of confidentiality, privacy, consent, damage of data that data can do. And I just want to talk about that a little bit. First of all, there's a standard trade-off between, anytime you're using, you know, human clinical data, right? There's a trade-off between trying to make it as widely available as possible so we can actually make medical breakthroughs, and the risks and harms that come with making it as widely available as possible, right? And one of the proposed solutions, and the reason I'm really bringing this up in this context, is we are the people who can start to think about computational solutions to these problems, right? And this is a meme of Larry Hunter's, if you will, but there are a lot of methods in cryptography that one can use to try to facilitate use of data without abuse of data. Most of them involve munging the data in some way. Most of them, therefore, limit the utility of the data to specific envisioned applications, and they're very application-specific and condition-specific. But I think I would like to get people in this room to think about these questions because they're really important to making sure that the field can move forward without major crises of confidence that just say, okay, we're going to lock it all down. It's not even ethical to do these kinds of studies. The other question that I have to bring up, even though I really wish I didn't, is that of consent. And who is giving consent for what use of their data and what is being done with it. So I guess we're going to play this clip, maybe. I will. Yeah, so this is...
And look, you might not care if a company wants to toss your data in a group called Kids and Cabernet so marketers can more effectively sell you things that make you seem like a bad mom in a fun way. But there is also a dark side here, because some companies can and do draw up even more narrowly targeted lists, like people with certain ailments or sexual preferences, and then sell those lists to anyone who wants to buy them. And what they can buy is pretty troubling.
WRAL bought thousands of names and addresses of local people with serious illnesses. This group living in the 27607 zip code have diabetes. These people in 27608 have cancer. These residents of 27609 have high blood pressure. And all of these locals battle depression. This list is moms to be.
So we bought this data and it tells us you're pregnant.
Yes,
And you are.
I am. Yeah, I am 18 weeks.
That's pretty creepy, isn't it? I honestly did not think there could be a worse thing to ask a woman you don't know than, are you pregnant? But you are pregnant, want to know who I paid to find out, has certainly entered the chat. And if you're thinking, but that's illegal under HIPAA, right? Well, no. As one researcher pointed out, the medical information that you relay to your physician is highly protected, but if you go to a medical website and search for terms like HIV or abortion, that information is not protected at all.
Okay, so I was supposed to stop there. So I guess the question, right, I don't think anybody in here is surprised by any of that. And at some point when you have free time, like on the flight back, watch the rest of this video because it's pretty funny. It's 25 minutes of John Oliver talking about what people can do with your data. But the upshot is even my undergrads who, you know, are studying computer science and data and biology were a little surprised by this. what do you suppose the general public thinks you could do with all this information, right? Like, this is not generally known. When we get consent forms signed, do they think that you can re-identify things using the kind of data that we're talking about using in mining? You know, I'm guessing not. And I'm guessing that even when we make really good faith attempts to obtain a informed consent, we're not probably doing the best job of making people informed. And there's a tension there as well, right? Because we want to be able to do these studies. It's important to do these studies. So again, I think computational solutions to all of this are things that we need to be talking about. Maybe not solutions per se. I mean, there are regulatory solutions to exploiting public data for personal profit and gain, but even using them for just, you know, more altruistic reasons, there are still problems, and we'd like to start to think about some of those. So with that, a couple of other topics that we might want to talk about include things like single cell data or spatial data that we've talked maybe a little bit about in this meeting, but there's a lot more to it. Accessibility versus privacy, and splicing, which we also haven't talked about in this meeting. And again, there's a whole other field of how do you deal with that? What do you do when you have multiple splice forms of something in a population? So they're sort of at different levels. I think that's kind of what we wanted to raise as questions related to these specific topics. And of course, we'll have broader discussions. So let's just get everybody up here from this group and we'll talk about, we'll take some questions.
Can we please use the mic for any questions? And we have about 14 minutes.
So that was amazing. I have so many questions, but I wanted to start with the one that's freshest on my brain, which goes into the question of drug repurposing. And I think it's a fantastic idea, and it's one of the things that really drove me to computational biology and the career path that I have as opposed to working for pharma. But I was wondering sort of the relationship between, one of the things we look at in cancer research is drug synergy, right? So we have drugs that work and then the cancer mutates. So I was wondering from the network, like how similar are those two problems? Are they similar at all? Are they totally different? You know, I had always wondered, do I look for networks between if I can build a network of drug response, are they closer together or not? And I have hypotheses about this, but I was wondering what your thoughts were.
So when you're trying to work with drug combination, you often want drugs that they are not hitting the same pathway, so they can activate two different pathways from the same disease. We have a hidden slide on that because otherwise it would be like way too long.
Hidden slides with this interface, so that's another piece. But
yeah, but what you often want is drugs that they are hitting different pathways, so they can work in a synergistic way and give you whatever the outcome you're looking for.
Yeah. And actually, I want to add something that we have a bunch of work out of our group and Lenore Cowan's group on. And many other people who have connections and tendrils here on between pathway models, right? Finding parallel pathways that are targetable, can complement to each other.
But follow-up question, I think given our, lossy knowledge of pathways and the fact that cancer drugs are dirty,
right? So you might have to develop them in a patient-specific way, right?
Yeah.
So you'd actually find which parallel pathways are in existence in those patients.
Yeah, I just want to bring up something that it just said, like the targets, right? So it is a major problem also with target discovery, because we have like about 10,000 genes that are known to be targetable. However, we only target like less than 2,000. So we have like this massive lack of study on how can we target those genes, how they can be really used, and that would be more like in the drug discovery setup instead of in a drug discovery. You go.
Let me add also one more thing, especially for cancer, where we really cancer may by itself break some pathways, which makes it easier for us, so we can use the understanding what are the complementary pathways, and then hit the complementary pathways with this drug, and then this is really perhaps the most desired situation because that drug will kill only cancer cells that have already something damaged. So, we have a lot of examples of this type of approaches, and this is one way to also looking to, use this not drug-drug interaction, but interaction with what's already damaged to look at what would be complementary things that can be hit. Yeah.
That was a great overview on so many different topics. I wanted to pick up actually on the first discussion point here because on single cell. Several of you, and certainly Teresa and Donna, you talked about a lot of the classic work on differentially expressed modules, and that was all from bulk gene expression, right? In the past few years, everyone's all about cell types and differences in cell type composition, and that being the driving signal as opposed to sort of gene expression. And so I wonder what your perspective is on, how much of that old work was really just confounders and how much of it was picking up on real signal, and have people gone back and kind of redone this with single cell, and are we going to see different things?
So I guess single cell is relatively new. So one thing, for example, that we look at it is we look at the pathways that are dysregulated in lung cancers, or genes that are overexpressed in lung cancers. And then once you start to get the grasp, that lung is a complex tissue, you start to ask the question, what are those? And you understand what are the markers of those tissue. You start to say, oh, we have mucin 5 going up. That tells me that basal cells are going up. And then using the single cell type of analysis, we can take bulk data, and there are methods, and there are new methods hopefully coming up that will help you to decompose the bulk data into cell type specific data and help to see how the cell types, for example, one thing that is being observed that with the smoking, you have decrease of ciliated cells and increase of other cells. So once you have a combination of single cell data and bulk data, you can make those inference. And some of this you could really observe based on the cell type specific markers. So that's one way. And we're not going to throw away all the bulk data, right?
Well, I think it's actually, most of it is still very plausible and accurate, but you're right that some of it is masking. You don't know whether it's changes in expression or changes in cell population.
Right, and many of them is changing in the cell population that we can now appreciate more than we could before. Yeah. I mean, at least for the lung cancer. So I think this is still quite new, but all these cell type atlases are very helpful to understanding how we can decompose. And there's also more things, also we cannot have cell atlas for everything, because if you have, say, really response to a disease status, you may have whole trajectory that you have, a cell moving from one type to another, and you observe everything in between. So there's a lot of, and that thing is probably should be one thing that we could also address looking from the single cell. And it's also things that we observe looking at mutation signature and so on, is the impact of immune system. That's related probably to immune infestation, and we can see also in cues like looking at the mutation signature, we look at the control samples, and we can see the relation, say, with apobex signature with the immune response. So there's a lot of things that we are just starting to look at, and I think it's very, very important, but I think there's lots of things to come.
Yeah, I mean, the immune response is a really interesting one for the bulk,
So
because
also from the image analysis
and in the cancer subtyping, actually, in some cancer types, some of the subtypes that were picked out from bulk were really just sort of distinguishing patients that had different levels of immune infiltration.
Yeah, and now when we can decompose the sample into various immune cell types, even that gives us some better understanding what might be happening.
The spatial models, especially integrating that into
and
will help.
Yeah, I think for box cell, right, we can see everything. For single cell, we can actually information, like we can do the life cycle right? So now, recent years, many people talk about life cycle. So a lot of subtype, they could be, they are different in this life cycle. So we can use single cell get complement information. But I think that's a very good point. This is very new data, new technique.
And new challenges, because we all know that single cell data are noisy. It's hard to get the genes that are not so highly expressed, and those tend to be transcription factors that are really important. So there's, I think, a lot of new opportunities for us to dive into this type of analysis.
So I have a question about the accuracy of patient-specific networks. So, I mean, we know inferring networks is hard, even with tons and tons of data, lots of samples. So what can you say about what is the approach of these patient-specific networks, and how do we know those networks are actually right? How much does that mess up the results?
I think that was the, how do you validate?
That was a great question, right? So again, patient-specific networks are a relatively new thing, and the answer to that is we don't yet know how well we know. I mean, we are starting to think about how do you validate. The short answer is they're proposed. I mean, so, the paper that I mentioned in passing in the beginning that does do these patient-specific networks in prostate cancer recommended a particular therapeutic approach to cells from, and so you can take these cells from these patients or models and do something in vitro to test efficacy on the cells. And basically, it looked like it was working for half of them, right? So is that good? I don't know, right? It's cancer drugs work and don't work in patients, and we still don't. I don't know what the control or what the background level of success is for the group, and it was a tiny number of samples in that group. So, 50% could be 25% or 75%, because I think it was 4 samples and it worked in 2, right? So, I have no idea what those numbers are. The short answer is those are the questions we need to be asking, is how do we actually validate this? And I can't answer that in a specific way, but yeah.
Sorry, just a related question. Because, there are probably 2 ways to think about patient-specific networks. One is that whether the networks are accurate or network is just some vehicle to some final endpoint that we really care about. So it's just a tool. So
it's really if the conclusions are accurate, not if the whole network is accurate. I'm sure the whole network isn't perfect, right? But I don't care.
So if it's the second thing, then the question is really, what does the network bring to the table, right? Could you have come to similar conclusions just using some high-dimensional, data
it doesn't
analysis?
sound like it, because this was a new therapeutic approach for prostate cancer in general. So, could you have come to those conclusions some other way? Maybe. Had anybody done so? No. But the open question is sort of how valid are those conclusions? And I think the point of the paper is somebody should go do that experiment. So they brought it to this point, but I'm sorry. Yeah.
Can you just tell us a little bit how we make patient-specific networks? I could envision multiple ways. Okay. One more time. Can you explain how we make patient-specific networks?
I mean, you can ask me how I would do it or how these other people have done it, right? I think the answer is there isn't a standard way to do it yet. They were looking at correlating, and again, I don't know if this is all of it, but at correlating attack-seq, open chromatin peaks and expression or downstream expression. And, there were a couple of other papers referenced on how they actually built the networks. So again, there are different approaches to doing that. Do I believe that that's all of that would work, all of it, to do what they did? No. So I'm sure there are other details that I haven't dug into yet. But
yeah, we work on autism, and so I doubt that this can become possible. We work on different types of data, from brain data, from brain span data set and things like that, where you can find, a lot of sort of neutral data, and that's your one network. But in many diseases, this could be difficult. It might be cancer related.
I mean, tissue specificity is another question. In autism, people work a lot with fibroblasts, which are at least accessible, but then there's a question of how well does that capture things in the brain. I'm doing some work in maternal immune activation where we're looking at accessible cells in the placenta as a model for the brain immune response in microglia. Because obviously people don't like you sampling microglia of their baby. So if you could do something useful with placental cells, that would be a good thing. So in general, you're right that it's not clear you can get the right network for the right patients. The other paper was looking at SNPs from sequencing, I think, blood, and mapping that per patient, and integrating it with existing data using methods we've talked about yesterday. So does this work? I don't know. Does it work in a way that is it a model that is useful for finding drugs is really the question. Because again, I'm sure you can poke holes in the network. So there's somebody behind you.
Last question for before the coffee break, but remember that we have discussion section after the coffee break. Mona.
Oh, yeah, I just wanted to mention one other challenge that I see with precision medicine and networks, which is the question of like equity and making sure that the methods that we develop actually work for people of different ancestries.
Okay, and like the $2 million personal clinical trial isn't like an equitable thing, you think?
Just questions surrounding that. Like, for example, things that, we can't, you would think some of this is just things that don't affect us, but actually, like, we use data, for example, that's based on reference genomes or reference proteomes. And so even methods that you develop for predicting interactions or whatever, they can be biased by the reference data sets. And so somehow, yes, and so we should also be thinking about like algorithmic approaches to doing targeted data collection or things like that that may, help with equity issues.
Yeah.
And
definitely.
just to follow up on that, the PMI has become All of Us initiative, and they just released a slew of data mining supplements for anyone with basically an R01, where they have made very conscious efforts to reach out and gather data from underrepresented research communities. It's only genetics right now, so I've been trying to find out if there's proteins, but I think there are. Again, if you have methods, I think that would be an incredible data mining opportunity to go into this data set and really identify specifically where the gaps are, because I think you're absolutely right. We're totally blind to what we're not seeing.
Thank you. Yes, that's really huge.
Yeah.
We're missing large chunks of the population.
Yeah.
And we know that, and we're trying to collect them, but it's not there, and it's not equitable. Yeah.
Let's end this session by thanking Daisy and the team, and then we're going to have plenty of time for additional discussion later.